Welcome to Biochemistry. I am Don Paras, your personal student teacher, here to educate you the metabolic processes of enzymes. In this lesson, we will be addressing the metabolic processes enzyme modeling, specifically the enzyme of sucrase. Now, let's go over some definitions. Enzyme: proteins that are a biological catalyst. Enzymes play an important role in influencing or expediting chemical reactions. The reaction encourages a lock and key method, also known as induced fit. Substrate: a substance or molecule the enzyme reacts with. Active site: a specific surface area on the enzyme on which the enzyme binds with a specific substrate. The reaction where an enzyme and substrate bind together to form an enzyme-substrate complexes. Cofactors and coenzymes: molecules that enhance with enzymes to work appropriately. They may bind permanently or temporarily on the enzymes. However, coenzymes are organic molecule carriers that hold an electron to bring it to another enzyme. Cofactors simply facilitate enzymes in catalyzation. Allosteric site: a specific region where a non-substrate molecule may bind onto an enzyme, thus determining whether an enzyme activates, inhibits, or turns off the enzyme. Allosteric activator: a non-substrate molecule that is binded with the allosteric site to activate and stabilize the enzyme functions, turns on enzyme to bind with substrate. Allosteric inhibitor: a non-substrate molecule that is binded with an allosteric site to inactivate the enzyme functions, turns off enzyme to bind with substrate. Competitive inhibitor: a non-substrate molecule that binds onto the enzyme's active site to disturb or inhibit substrate molecules to bind onto the enzyme. Non-competitive inhibitor: a non-substrate molecule that binds to a region on the enzyme, specifically not on the active site, thus change the conformation or shape of the enzyme. The original substrate may no longer bind to the enzyme as the enzyme's identity has changed. Reaction with sucrase: sucrase is an enzyme that helps break down the disaccharide molecule of sucrose to glucose and fructose molecule. The reaction that takes place during the breakdown of sucrose is hydrolysis, as water is added and H goes to one molecule and OH goes to another molecule. The sucrase enzyme simply binds with the sucrase substrate to break it down to glucose and fructose. The sucrose will be reacted with the water with the help of the sucrase catalyst to produce glucose and fructose. In this model, we have an enzyme of sucrase and a substrate of sucrose. Under the condition of collision theory, they will meet at the activation point, where the sucrose will now turn into fructose and glucose that will then be used to the body. The main function of sucrase can always be reused, and it plays an important role into helping the body digest sucrose. Sometimes enzyme needs some assistance, especially when the activation point is not complete with the substrate. As you can see here, the activation point do not meet the substrate, thus it will need a cofactor that will connect itself to the activation site, and then under the condition of the collision theory, when they connect on the activation point, they can now break down into glucose and fructose. Thus, cofactors play an important role into helping the enzyme break down big macromolecules. Now here is the overview of the enzyme model. The top part over here is the allosteric site. The bottom over here is the activation site. Both of these sites play a specific role. Now here is an allosteric inhibitor model. The allosteric inhibitor will connect itself to the allosteric site. It will encourage the activation site to enclose. At this point, the sucrose or the substrate could no longer bind with the enzyme. Now here is a model of the allosteric activation. The allosteric activation will connect itself to the allosteric site. The allosteric site will communicate to the activation site to open. Thus, the substrate can connect itself to the enzyme and can break down to the molecules that it must be. So the sucrose and sucrase will turn the sucrose into fructose and glucose. Now here is a model of a non-competitive inhibitor. The non-competitive inhibitor will bind itself onto the enzyme. The enzyme will change its identity, and the substrate can no longer bind with the enzyme. Note that the non-competitive inhibitor cannot bind itself onto the activation site. Here is a model of the competitive inhibitor. The competitive inhibitor will bind itself onto the activation site. This will prohibit the substrate to bind itself onto the enzyme.